In the summer of 1848, a French agricultural chemist named Jean-Baptiste Boussingault sealed a young bean plant inside a glass bell jar, fed it nothing but distilled water and carefully measured volumes of air, and waited. He was not the first person to attempt such an experiment. Nearly 2 centuries earlier, a Flemish physician had tried something similar with a willow tree and a weighed pot of soil, and the result had been so confounding that the scientific establishment spent generations arguing about what it meant. But Boussingault had tools his predecessor lacked: a precise analytical balance, reliable methods for measuring gaseous exchange, and a conceptual vocabulary that included the word carbon dioxide, a compound that had not been identified when the earlier experiment was performed. When Boussingault opened his bell jar and weighed his plant, the results confirmed what a small number of chemists had begun to suspect, but what the broader world still found almost impossible to accept. The plant had gained mass. The water could account for some of it. The soil could account for almost none of it. The overwhelming majority of the new material in that plant had come from the air. This finding was not new in 1848. Its essential outlines had been emerging for nearly a century through the work of Joseph Priestley, Jan Ingenhousz, Nicholas, Theodore de Saussure, and others. But it remained, and in many ways still remains, one of the most psychologically resistant facts in all of science. The notion that a tree, an object that can weigh 40 tons and stand for 1,000 years, is constructed primarily from an invisible gas that constitutes less than 1/20th of 1% of the atmosphere, contradicts every intuition the human mind brings to the question. We see the tree rooted in soil. We see the soil displaced by expanding roots. We see earthworms and fungi and decaying leaves feeding the ground beneath the canopy. The narrative writes itself. The tree eats the earth becomes the tree. It is a story so visually coherent, so consistent with our daily experience of watching things grow from where they are planted, that correcting it requires not merely new information, but a fundamental restructuring of how we think about the relationship between visible matter and invisible substance. The original experiment that began to unravel this misconception was conducted by Jan-Baptist van Helmont sometime around 1630, though it was not published until after his death in 1644. Van Helmont was a physician, a mystic, and an alchemist working in Brussels, a man who occupied the borderland between medieval and modern thinking and who brought to his investigations both the rigor of measurement and the speculative enthusiasm of a tradition that believed in the transmutation of elements. His experiment was, by the standards of his era, a masterpiece of controlled observation. He dried 200 pounds of soil in a furnace, placed it in a large earthenware pot, planted a willow shoot weighing 5 pounds, covered the pot to prevent contamination by dust, and watered it exclusively with rainwater or distilled water for 5 years. At the end of the experiment, the tree weighed 169 pounds and 3 ounces. The soil, when dried again, had lost only 2 ounces. 164 pounds of tree had materialized, and the soil was essentially unchanged. Van Helmont concluded, reasonably given his conceptual framework, that the tree was made of water. The water had been transmuted into wood. This was wrong, but it was wrong in an instructive way, because it demonstrates how the absence of a single concept, in this case, the concept of a gas as a substance with measurable mass that can participate in chemical reactions, can prevent even a brilliant experimentalist from interpreting his own data correctly. Van Helmont could not see the air as a source of raw material, because the air, to a 17th century thinker, was not a material in the way that soil and water were materials. It was a medium, a void filled with nothing of consequence, the space through which birds flew and winds blew, and through which you could wave your hand without encountering resistance. The idea that this insubstantial nothing could be the primary ingredient in the construction of solid wood was in the intellectual context of the 1600s, approximately as plausible as the suggestion that a house could be built from silence. The slow unraveling of this misconception tracks almost perfectly with the development of modern chemistry. In 1727, Stephen Hales, an English clergyman and physiologist, published a book called Vegetable Statics, in which he demonstrated that plants exchange large volumes of air through their leaves. Hales did not know the composition of this air or the nature of the exchange, but he established quantitatively that the atmosphere was not merely surrounding plants, but passing through them in significant quantities. In 1771, Joseph Priestley, working in Leeds, demonstrated that a sprig of mint could restore air that had been rendered unbreathable by a burning candle or a suffocating mouse. The plant was doing something to the air, restoring some quality that combustion and respiration had removed. Priestley did not understand the chemistry, but he had identified the basic transaction. Plants give back to the atmosphere what animals and fire take from it. In 1779, Jan Ingenhousz, a Dutch physician living in London, refined Priestley's findings with a series of experiments that demonstrated that this restorative process occurred only in sunlight and only in the green parts of the plant. In darkness, plants behaved like animals, consuming the good air and producing the bad. The green leaves were doing something in sunlight that reversed in darkness. Ingenhousz was circling the truth, constrained by the phlogiston theory that still dominated chemistry, unable to name the gases involved with the precision that would come later, but identifying the essential parameters of a process that would eventually be called photosynthesis. The final pieces fell into place through the work of Antoine Lavoisier, who dismantled the phlogiston theory and established oxygen and carbon dioxide as distinct chemical compounds, and Nicolas Théodore de Saussure, who in 1804 published the first quantitative account of plant nutrition that correctly identified carbon dioxide and water as the principal inputs, and oxygen as the principal output. De Saussure measured the carbon content of plants, the carbon dioxide content of the air they were exposed to, and the change in both over time. His numbers were clear. The carbon in the plant came from the carbon dioxide in the air. The hydrogen came from water. The oxygen released by the plant came from the splitting of water molecules. The soil contributed minerals, essential, but constituting only a small fraction of the total mass. 2 centuries later, these facts are not in dispute. They are confirmed by isotope tracing, by mass spectrometry, by radiocarbon dating, by every analytical technique that modern chemistry has devised. When you measure the elemental composition of a dried tree trunk, you find that it is approximately 50% carbon by mass, 44% oxygen, and 6% hydrogen, with the remaining fraction composed of nitrogen, phosphorus, potassium, calcium, and trace minerals. The carbon came from atmospheric carbon dioxide. The oxygen came partly from carbon dioxide and partly from water. The hydrogen came from water. The minerals came from the soil. Added up, and you find that roughly 90 to 95% of the dry mass of the tree was sourced from the atmosphere and from water, not from the ground. The mechanism by which this occurs is photosynthesis, and while the word is familiar, the process itself is of a complexity that rewards close attention because it is doing something that no human technology has managed to replicate at scale. And the details of how it accomplishes this illuminate not just botany, but the deepest questions of thermodynamics, quantum mechanics, and evolutionary biochemistry. The process begins at the surface of a leaf. The outermost layer of a typical leaf is the epidermis, a single layer of transparent cells coated with a waxy cuticle that minimizes water loss. Scattered across the epidermis, primarily on the underside of the leaf in most species, are the stomata, microscopic pores, each flanked by a pair of kidney-shaped guard cells that can swell or shrink to open or close the pore. A single square centimeter of leaf surface may contain tens of thousands of stomata, and through these pores the tree conducts its most essential commerce with the atmosphere. Carbon dioxide enters, oxygen and water vapor exit. The regulation of stomatal opening is itself an extraordinarily sophisticated process, governed by light intensity, carbon dioxide concentration, humidity, temperature, and plant hormones, balancing the need to acquire carbon against the risk of losing water in a calculation that the plant performs continuously, without anything that could be called a nervous system, using purely biochemical signaling. Inside the leaf, the carbon dioxide molecule diffuses through the intercellular air spaces and dissolves in the thin film of water that coats the surfaces of the mesophyll cells. From there, it enters the chloroplasts, the organelles where photosynthesis takes place. Chloroplasts are roughly the size of bacteria, and this is not a coincidence. The endosymbiotic theory, now supported by overwhelming genetic evidence, holds that chloroplasts are the descendants of ancient cyanobacteria that were engulfed by a larger cell more than 1 billion years ago, and that, rather than being digested, established a symbiotic relationship with their host that persists to this day. Every chloroplast in every plant on Earth retains its own small circular genome, a vestige of its free-living ancestor, and it replicates independently within the cell, dividing by fission in a manner reminiscent of bacterial reproduction. The tree, in a sense, is not a single organism but a community, its photosynthetic capacity provided by the descendants of microbes that surrendered their independence in the Proterozoic eon and have been manufacturing sugar for their hosts ever since. Inside the chloroplast, the process unfolds across two distinct but coupled stages. The first stage, the light-dependent reactions, takes place in and across the thylakoid membranes, an elaborate system of internal membranes folded into stacked discs called grana. Embedded in these membranes are the photosystems, large protein complexes containing hundreds of pigment molecules, primarily chlorophyll A and chlorophyll B, along with carotenoids and other accessory pigments. These pigments absorb photons of specific wavelengths, primarily in the blue and red portions of the visible spectrum, and funnel the captured energy toward a special pair of chlorophyll molecules at the reaction center of each photosystem. What happens at the reaction center is, at the quantum level, one of the most remarkable events in biology. The energy of the absorbed photon excites an electron in the reaction center, chlorophyll, to a higher energy state. This excited electron is then transferred to a nearby electron acceptor molecule, initiating a chain of electron transfers known as the electron transport chain. The electron moves from carrier to carrier, losing energy at each step, and the energy released at certain steps is used to pump hydrogen ions across the thylakoid membrane, creating an electrochemical gradient. This gradient drives the enzyme ATP synthase, a rotary molecular motor that synthesizes adenosine triphosphate from adenosine diphosphate and inorganic phosphate. The electron ultimately arrives at a molecule of nicotinamide, adenine dinucleotide phosphate, reducing it to its energy-carrying form. The electron that was removed from the reaction center chlorophyll must be replaced, and this is where one of the most chemically demanding reactions in all of biology occurs. The enzyme complex known as the oxygen evolving complex, a cluster of 4 manganese atoms, 1 calcium atom, and 5 oxygen atoms, held in precise geometric arrangement by surrounding protein scaffolding, catalyzes the oxidation of water. It rips water molecules apart, extracting their electrons to replenish the reaction center and releasing molecular oxygen as a byproduct. This reaction requires the accumulation of 4 oxidizing equivalents, meaning that the complex must absorb 4 photons and remove 4 electrons from 2 water molecules before it can release 1 molecule of oxygen. The chemistry involved is so energetically demanding that no chemist has yet succeeded in building a synthetic catalyst that can perform it with comparable efficiency, despite decades of effort motivated by the obvious applications in clean energy technology. The oxygen released by this process is, from the tree's perspective, metabolic waste. From the perspective of nearly every animal on Earth, it is the prerequisite for existence. The breathable atmosphere is a byproduct of plant metabolism, a waste stream that happens to be the single most important chemical resource for aerobic life. Before the evolution of oxygenic photosynthesis, approximately 2.5 billion years ago, the Earth's atmosphere contained negligible, free oxygen. The organisms that evolved this capability, the cyanobacteria, ancestors of modern chloroplasts, transformed the planet so thoroughly that the event is recorded in geological strata worldwide as the Great Oxidation Event, a boundary in the rock record that marks the transition from an anaerobic world to an aerobic one. The rusting of iron deposits, the formation of banded iron formations, the mass extinction of anaerobic organisms that could not tolerate the new reactive atmosphere. All of these are consequences of what is at root. The same chemical reaction that occurs in every sunlit leaf on every tree in your neighborhood. The second stage of photosynthesis, the Calvin cycle, uses the chemical energy produced by the light reactions to fix carbon dioxide into organic molecules. The key enzyme is ribulose -1, 5-bisphosphate carboxylase, deoxygenase, universally known as Rubisco, a molecule so central to life on Earth and so spectacularly inefficient at its job that it deserves particular attention. Rubisco catalyzes the attachment of a carbon dioxide molecule to a 5-carbon sugar producing two molecules of a three-carbon compound. This is the step at which inorganic carbon becomes organic carbon, the step at which air becomes wood, the step at which the invisible becomes solid. It is the most important chemical reaction in the biosphere by mass. Rubisco is also, by the standards of enzymatic biochemistry, agonizingly slow. Most enzymes catalyze thousands or tens of thousands of reactions per s-econd. Rubisco manages approximately 3. It also has the unfortunate tendency to confuse carbon dioxide with oxygen, catalyzing a competing reaction called photorespiration that wastes energy and releases carbon dioxide rather than fixing it. This competing reaction is sufficiently costly that many plant lineages have evolved elaborate workarounds to minimize it, including the C4 photosynthetic pathway used by grasses and many tropical plants, and the crassulacean acid metabolism used by cacti and other succulents. These adaptations concentrate carbon dioxide near Rubisco, reducing the probability of the wasteful oxygenation reaction. The very existence of these complex evolutionary innovations is a testament to Rubisco's limitations, a molecular hack built on top of an ancient enzyme that has never been replaced because it was there first and because evolution, unlike engineering, cannot redesign from scratch. Through the Calvin cycle, the three carbon products of carbon fixation are reduced, rearranged, and ultimately combined into 6 carbon sugars, primarily glucose. From glucose, the tree synthesizes the full range of organic molecules it needs. Sucrose, the transport sugar, carries energy from the leaves to the rest of the tree through the phloem. Cellulose, the structural polymer, forms the walls of plant cells and constitutes roughly 40 to 50% of wood by dry mass. Lignin, a complex and irregular polymer of phenylpropanoid units, fills the spaces between cellulose fibers, providing rigidity and resistance to compression, and making wood the remarkably strong and durable material that it is. Hemicellulose, pectin, proteins, lipids, nucleic acids, the entire molecular inventory of the tree, all ultimately trace their carbon back to carbon dioxide molecules that entered through the stomata and were fixed by Rubisco. The scale at which this process operates is difficult to appreciate intuitively. A large oak tree may have 200,000 leaves, each with tens of thousands of stomata, each stoma admitting carbon dioxide molecules at a rate determined by atmospheric concentration, wind speed, temperature, and humidity. Over the course of a single growing season, a mature deciduous tree in a temperate climate may fix between 20 and 50 kilograms of carbon, pulling that carbon entirely from the atmosphere. Over a lifetime of 200 years, such a tree may accumulate several metric tons of carbon in its trunk, branches, and roots, all of it sourced from the air. A hectare of mature forest stores between 100 and 400 metric tons of carbon in its biomass and soil, depending on the forest type and climate zone. The global total, the amount of carbon currently stored in the world's forests, is estimated at approximately 861 billion metric tons, an amount roughly equivalent to a century of current global fossil fuel emissions. These numbers transform the seemingly academic observation that trees are made of air into a matter of planetary consequence. When a forest is cleared and burned, the carbon that was stored in those trunks and roots and soils is returned to the atmosphere as carbon dioxide. When a forest is allowed to regrow, atmospheric carbon is pulled back down and locked into wood. The global carbon cycle, the system of reservoirs and fluxes that determines the concentration of carbon dioxide in the atmosphere, and therefore the trajectory of the Earth's climate, depends critically on the photosynthetic activity of forests, grasslands, and other primary producers. The balance between what is pulled down and what is released determines whether atmospheric carbon dioxide rises or falls, and for most of the last 10,000 years, that balance was roughly in equilibrium. It is no longer. But this is not merely a story about carbon accounting and climate policy, as important as those topics are. There is something more fundamental at work here, something that touches on the deepest questions about what matter is and how it moves through the world. The carbon atom sitting in the heartwood of an oak tree has a history. It did not come into existence when the tree grew. It has existed since it was forged in the interior of a star billions of years ago through the nuclear fusion of helium nuclei. That carbon atom was ejected into space when the star exploded, drifted through interstellar clouds, was incorporated into the accretion disk that formed the solar system, became part of the Earth's crust or atmosphere, and has been cycling through geological and biological systems ever since. It may have been part of a limestone reef in the Ordovician. It may have been dissolved in the deep ocean for 50 million years. It may have passed through the body of a dinosaur, been exhaled, been fixed by a Cretaceous conifer, been buried in sediment, been compressed into coal, been mined and burned and released back into the atmosphere, and been captured again by the oak tree in your backyard. The biography of a single carbon atom spans the entire history of the planet, and before that, the life cycle of a star. This is what Feynman meant when he pointed at a tree and said it came from the air. He was not merely correcting a botanical misconception. He was illuminating a chain of causation that connects the nuclear physics of stellar interiors to the biochemistry of a chloroplast, to the growth rings of a tree in a California garden. He was pointing out that the solid, heavy, manifestly tangible substance of wood is, in its origin, as insubstantial as a breath, as weightless as a molecule of gas, as invisible as the carbon dioxide that you are exhaling as you read this. The tree is a monument to transformation, a demonstration that the boundary between the substantial and the insubstantial is not fixed but fluid, that the same atoms can be air one year and wood the next. That solidity is not a permanent condition of matter, but a temporary arrangement maintained by the continuous input of energy from a star. There is a passage in the writings of the physicist Erwin Schrödinger from his 1944 book, What is Life?, in which he attempts to describe how living organisms maintain their complex organization in the face of the second law of thermodynamics, which demands that all systems tend toward disorder. Schrödinger's answer was that living things feed on negative entropy, that they import order from their environment and export disorder in the form of waste heat and degraded molecules. A tree does exactly this. It takes disordered carbon dioxide molecules from the atmosphere, which are low in chemical potential energy, and assembles them into highly ordered cellulose and lignin polymers, which are rich in chemical potential energy. The energy that drives this ordering comes from sunlight. The entropy cost is paid by the sunday, which generates enormous amounts of disorder internally for every photon of ordered light it emits. The tree is, in Schrödinger's framework, a local island of increasing order in a universe that is, on the whole, running down. It is a temporary victory over entropy, sustained by the continuous expenditure of solar energy, and it will be reversed when the tree dies and decomposes and its carbon returns to the atmosphere. This cycle, growth and decay, fixation and release, order and disorder, has been running on Earth for approximately 470 million years. Since the first land plants colonized the barren continents and began pulling carbon from an atmosphere that, at the time, contained carbon dioxide concentrations many times higher than today's. Those early plants were small and simple, lacking true roots or leaves, but they were already performing the fundamental chemical trick that all their descendants would inherit. They were turning air into body. They were constructing themselves from the invisible. Every tree you have ever seen is doing the same thing, right now, in silence, without effort, without instruction. The canopy above you is a factory floor where carbon dioxide is being disassembled and rebuilt, where sunlight is being captured and converted, where the insubstantial is becoming substantial at a rate of millions of molecules per s-econd per leaf. The water feeds the reaction. The minerals provide essential cofactors. But the mass, the sheer physical presence of the tree, the thing that makes it a tree and not a reed or a fern or an empty space, came from the air. It was always the air.